we do at home. And people are tied with ropes to that snowmobile and to everybody else. And such that if somebody were to have an accident, no, they're tied to things. And so there are two people who have short leashes, which are in front, and then two people who have long lines that are further in the back to keep that. But no, as long as we take a radio and we let on the board that we're going up the glacier, you can go up to the glacier any time of day and night because we have 24 hours of light. So it is just as light now as it will be at 11 p.m. It's a little bit darker, but between midnight and 2 a.m., but you wouldn't be able to tell the difference. So we can go up the glacier, and we can also walk around what we call the backyard, which is now this coastal rocky area along the water all the way to the edge of the glacier. And people there have tents, have platforms. They spend the night out with sleeping bags because otherwise everybody here is in a room with bunk beds. Little tiny rooms. One bunk bed, one desk, a metal closet per person, and there isn't a lot of privacy. And so if you want to have some quiet space or something, you either go walking along the rocks, and people, you will see them walk out with guitars, and then you see them singing and playing out in the rocks, or they simply just go out and spend the night with tents. There's one little, what they call rec house, in case you don't have a tent. And people at stations, since it's really not that cold, right? It's 32, 28, 20, but it's not that bad now with a sleeping bag and and spend the night out. A lot of people do that. Okay, I'm done.
Okay, Patty, it's 9:36 right now. Do you want to take one more question, or do you want to move inside?
I'll take one more question and then I'll move inside.
Okay, from the same room then. You have one more question you want to ask?
How many times, what were the things you unexpectedly didn't actually wanted to see, but it was unexpected, like something unknown that you didn't know?
Are you asking for her to share something she's seen that was unexpected?
Something unknown that none of the world actually even knew about?
I'm having a hard time hearing.
Or never actually knew about.
Have you found anything new?
Like new, like something that you didn't know, like something that's really you didn't even.
So did she find something new that was unexpected that she didn't know about before? Is that the question?
Yes.
Okay. Have you found something new that you didn't know about that was unexpected, Patty?
Oh, there are lots of things. So I'll give you one example. I went walking down the backyard because the rocks are open, and I could get to the very edge of the glacier. And then as I'm walking, I heard this sort of like a song, sing song, something that I hear when I'm in Maine hiking. When it's spring and it's melting and all the little rivers and streams and all start to be full of water, right? There's a song that they play as you walk down. Well, that's not what I expect here in Antarctica, because I always think that it's nothing but rock and frozen water and seawater. And here it is, this glacier is melting and the water is running just like a little stream. And underneath it, if you look into those, there's stuff that is starting to grow. In this case, it's lichen or something, others. And you can just start where it was brown rock. It's just now a little bit yellow. That was one. The other one that is really neat, it's sad, but it's really neat, is that there are what's called Glacier Strata. So marks that the glacier has left when it formed on the rocks, really like gouges, and the rocks are polished, smooth. You also see that in Maine because almost all of our rivers, are old glacier fjords, And so you see that some of the most spectacular ones, perhaps, Bar Harbor, but you see, and now these are all exposed. You can touch them, and you can see then how strong a force. You can see it, you can touch it. The glacier is when it's forming, and how sad it is that they are retreating so much. And I can go on. There's just watching some of the whales do some of their fishing was just spectacular. We were out in the Zodiac, and I promise, I was 10 feet away. And so for me, who studies small microscopic things, I'm just as mesmerized by big things like anybody else.
Are you all
I'm
set
done.
then? Okay, great. Well, thank you for your great questions. That was a great first round of questions. Patty, do you want to take us into the lab now?
Yeah, so this is what's going to happen. I'm going to pick up this computer, yeah, and the camera will still be on. And so it'll be jumbly and moving as I walk. I'm going to walk down all this stairs, and then go through the building, and then through the labs, and then all the way to where my lab is, so that you can see what we do all day, okay? So it'll take me about 3 to 4 minutes. So think about questions or something, or get up and stretch and move your arms while I do this, okay? So all of these wooden walkways are built, of course, for safety, so that in the winter time, because the station is open year round, you can transit from one place to the other in safety. Oh, I know what I can do. I can stop right by my blue box now that I'm on my way. There, can you see the big blue box now?
Now I see the big blue box.
Okay. So guys, this blue box here, which we brought all the way from the US, is where our experiments are. And so it's filled with bottles that have seawater in triplicate. There are 4 different treatments, and we'll call that an incubator. And so it keeps the water at the same temperature as in the ocean. It keeps the light similar to the depth in which we collected the water, which was 5 meters. And then it rocks the whole thing, so that it's similar to the motion that the phytoplankton experience in the ocean. If you remember from your cultures, right, those weren't really being moving, and then everything at the end, that would be a question I'll ask you. What happened to your cultures after several days? Where were all the cells? And so then you see all kinds of tubing going in and out. This is how we feed, we add the treatments that's being done, and those are being done on a continuous basis. And then this, anyhow, we'll leave it at that. Now I'm gonna go in. It's gonna get dark because I'm going through the aquarium room so as not to lose the connection. I'll turn the lights on. And so this is the aquarium room. It's full of tanks, which normally people who come and study fish, they use, but they won't come until March or April, okay? And so these here are some of the pumps that we're using to control the treatments. Well, this guy here is what controls the temperature of the water and the rocking of the cradle. And of course it broke. And that's one very important thing, is that when you come out into the field, you can see it's all taken apart now. And when you come into the field, you need to be prepared for everything breaking, and you have to bring, well, everything. as there is no store, there is no place to go and buy anything. Okay, we're going into the main labs now. And I'm just walking down the hallway. And here comes Dr. Conway, who is my co-investigator. You see, he's got a smiley, friendly face. Okay. Keep going. Let's get stable soon. Okay. All right, so here's the lab, and let me undress a little bit first because there's a slight difference in temperature here. I will keep the orange thing on so that you can tell me. No, there, yeah, okay. So there are about 10 labs, little labs in this big lab, and we get this space and we pack everything that we need. We actually packed and sent our things last August, even though we were coming in December. And so our things were shipped to California, and then from by truck, and then from California they were shipped by boat to Chile, to Punta Arenas, and they wanted things to arrive 2 months before we arrived, and then they were shipped by boat from Punta Arenas to Palmer Station a month before we arrived, except for just 3 boxes with more delicate instruments that came by air freight. But we ship everything from paper towels to beakers to chemicals to notebooks to computers to clothes, of course, no outerwear, to our special food. No, there's certain kinds of crackers that I like. Carlton, my technician here, whom you see him reading there, he's running samples on a gas chromatograph. I'll show you. He shipped like, I don't know, 10 pounds of coffee. Okay. So what did we come to do here in Antarctica? We came to study how bacteria and phytoplankton play together. And so if you think about for a second that we have bacteria in our bodies and that those bacteria are actually our friends, they keep us healthy. It's the same thing in the marine environment, whether it's a seal, a penguin, or phytoplankton and bacteria, and they talk to each other, just like our bacteria in our bodies talk to each other. When they're not talking to each other, or it turns out that it is a bacteria or a virus that is not friendly to us, we get sick. Same thing here. So you would say, well, why did you have to go so far to do that? Why couldn't you just do it in the Gulf of Maine? Well, there are millions of bacteria and millions of phytoplankton all over the oceans. But here in the polar regions, there are phytoplankton that make a certain compound. It has sulfur in it, okay? And so the bacteria respond to that. Because climate change is most felt in polar regions, we figured, okay, we'll come here, we'll look at this small group, let's see if we can see how they respond, and let's see if we can see if their genes or something changes. And then we can scale up to all of the phytoplankton and all of the bacteria. Now, remember, many of you did the phytoplankton cultures, and you did all kinds of experiments. Some of you did with light and without light, with nutrients and without nutrients, colder and warmer. And so when it's your turn to ask questions, I want you to tell me if you saw any differences. I know from some of you that you did. I also want you to tell me if the cultures were green all the time, or did the stuff tend to fall to the bottom of the flasks? Because that's important for what I showed you outside, okay? Okay, so with that, I'm gonna walk into the lab, okay? And so we have things like a humongous freezer. This is here. And then here is Carlton, who has computers. He can't hear me because he's listening to music. So I can say almost anything I want, and he won't even know that I'm talking about him. And so this instrument here is what we call a gas chromatograph. Yeah, and so we put samples in there and they tell us what compounds are there and how much. And so we brought all of this from Maine, okay? The gases, well, they brought those for me from Chile. It would have been too expensive to bring them from the US. Then we have a hood, and then, we pin everything up and down so we can communicate. Everything in here, from balances to water baths to pipettes. I guess Pete is ready to clean something in here, or he's measuring something. Okay. You see, you said if we have water, we have a sink. We actually can put seawater down this sink instead of just fresh water. And I'll explain why I said that. Antarctica is covered by a treaty. It is a protected region. And so everybody who comes needs to take with them everything they bring. And so all the chemicals that we use up here, we just don't dump them down the drain. They all get collected. And then there's a group of people here that all they do is waste management. All the paper, all the chemicals, everything. Some things get burned here. We evaporate as much as we can, but a lot of it will then end up on the boat, and it may end up in Punta Arenas in Chile. And if not, it will go all the way back to the US. When we arrived, the ship, when we came, they were doing every 3 years, they have this major cargo of hazardous waste, stuff that cannot be thrown. And that goes by ship to Punta Arenas. Then it goes on to the other ship, which then takes it all the way back to the US. So it's you have to be very careful with what you do with putting things into the environment. Okay, I've talked enough. I'm going to answer the question that why did I become a scientist, okay? And then we'll go back to the questions. I became a scientist because I had lots of questions, and because I had a very hard time doing things when I was told to do them. And so I figured in my wisdom as a young person that as a scientist, I would get to ask questions and find the answers by myself. Well, that's a little bit naive, because everything I do requires a team of people, whether it's in the science, in the lab, or whether it is when I am sharing my science with you, right? So we are a team too. You guys there in your classrooms are asking me questions, and I'm sharing with you. And then you ask me questions, which goes, I hadn't thought about that. And so everything that a scientist does is part of a team. And that's actually a lot more fun than just doing things by yourself, the way I thought at the beginning. Okay, Lisa, I'm done. Their turn.
Okay, so we have about 6 minutes left. I'm going to go reverse order through the rooms this time. And we'll start with one question per room, and we'll loop around until we run out of time. Okay, so let's see. The Dedham room, do you have any questions you'd like to ask? What's your top question right now?
How long are you in during the year at the lab? Like how?
Okay, let me answer that. Let me answer that in two different ways, okay? First, here in Antarctica, and I will be an answer to the question that was asked earlier. We left Maine on November 30th, arrived in Punta Arenas on the 1st, and arrived here on December 6th. And we will leave from here on February 6th and arrive back into the mainland on February 6th, February 10th. So we will have been door to door about a little bit over 2 months, okay? In this lab. At home in Maine, in Booth Bay, at Bigelow, I'm in the lab every day, except when I'm on travel. And as Lisa knows, I'm on travel a lot. Okay, I'm done.
Okay, thank you for your question. I'm going to move over to Ms. Westra's room. Do you have a question? Do you have a question? Yes, I see you have a question you'd like to ask.
Great. What's the question? You already asked me. Do you have your question? How do you guys sleep?
Oh,
You already
answered
This is
that.
about phytoplankton.
About phytoplankton.
Who has a question about phytoplankton?
I do.
Say it.
How long are you gonna be in it?
She just answered that.
Okay.
How do phytoplankton mate?
Was the question
How
how
phytoplankton mate?
Yeah.
Oh, that's a great question. So first of all, most phytoplankton are plants. And so they mate by just like plants on land do. In which you have flowers and then you have pollen, which is the male, and then you have the eggs, which is the female part. However, most phytoplankton simply divide. And only when they get really small, so once every 1,000 times or more, once they get really small, they then generate sexual cells, male and female, and form, in the case of diatoms, something called an oxospore. But most of phytoplankton just simply divide. Next question.
All right, Ms. Patnod's class, what's your question?
How much plankton do you only study plankton?
Yes, I only study plankton. However, among the plankton, there are animals, zooplankton, there are plants, phytoplankton, there are bacteria, bacterioplankton, there is viruses, viral plankton, and there's now something called archaea. So there is a lot of plankton, and among each one of those groups, there are thousands and thousands of species. So my work focuses on phytoplanktons, in other words, the microalgae, but because they interact with bacteria, I do a little bit of that as well. Because they're food for zooplankton, I do a little bit of that. And because virus will attack them, well, then I look a little bit at that as well. Next question.
Okay,
Thank you.
Ms. Venger's room, do you have a question?
Okay, so Oxygen 8 wants to know how many hours a day do you work? Are you on a 24-hour rotation or?
So here we are on a 24-hour rotation, and we work no matter just what's needed. But because we have this experiment, there are a couple of days in which we're up for like 18 hours. And then the other days we're waiting for them to grow. And so we're maybe doing the experiment part about 4 hours. So this morning I got up at 7:20. At 7:30 I was having breakfast. By 8 o'clock I had turned everything on. By 8:30 I collected my samples. This is why we're talking at this time. I then ran fluorescence, ran chlorophylls, and I went and talked to you. And so then after that I'll either look at data or may do something else. But last night I went to bed at 10 after I had prepped all the samples for Carlton to run this morning. So we don't have an 8 to 5. Some days are quieter than others, but we are up whenever we need to be up, whether it's Saturday or Sunday, but paying close attention that we don't get overtired. So we try to take a day off somewhere in there. Next question.
Okay, Ms. Train's class, what question do you have?
We were wondering, because looking at the big tank outside, the big blue tank that goes up and down, if you're not sampling the water in Antarctica, in the sea, how come you couldn't just take the Antarctica water and bring it to Maine and study it there? Why do you need to be on scene?
So the reason we have the tank, even with that tank, it's very hard to mimic the natural conditions. When I said that something broke inside, we have this air conditioning and cooling and heating system to try to maintain the water being at somewhere between -1 and +1 degrees Celsius. It's very hard. And so the water would get too warm on the way there. And then I also need to keep the light just the right way. And every time we try to get cultures like I sent you from here to Maine, and that's usually only maybe 48 hours, at least half of them die. Next question.
Okay, so it's 10 o'clock. Do you have time, Patty, to stay for one more round of questions?
Yes,
Okay.
I do.
Do any of the teachers unmute your microphones? Well, let's just, can you stay for a few more questions? Teachers, classrooms, you got one more round in you? All right, no one's getting thumbs up and nods. Okay, so I'm gonna go back around, back to Dedham. Do you have another question you'd like to ask in Ms. Tate's classroom? Anything in?
Let's see. Keep
Ms.
talking.
Tate?
Since we're going to do this, I'm going to take you to the front of the station while we ask questions. You wanted to see, since we're now in borrowed time, right? So I'll take you upstairs first so that they can see where we eat, okay?
Okay, great.
But they can keep asking questions in the meantime.
Yep, they will. Ms. Tate, do you have a question in your room? Nope.
No.
Oh, okay, great. So going back to Ms. Venger, do you guys have another question in that room?
Okay, yeah, let's see. Itrium wants to know about krill. Go ahead, ask.
Is there any krill there?
Is anybody working with krill?
Yes, there is. Give me 2 minutes. I didn't realize it was the beginning of lunch, okay? So I'm just going to give you a tour and I'm going to get out of here, okay? Right? Okay. All right. Now was having lunchtime. So here we are in the front of the station, and you can see the mountains in the back. That's the mainland now. And here, this roof, that's the boathouse. And then here is our little harbor. And then you can see some bigger pieces of ice, and which is a piece of glacier right there that used to be huge, okay? So I'm just gonna keep you here and answer your questions now, the next round of questions, okay?
So you had a question.
There you go.
There is someone working with krill at the station?
Yes. There is a group working with krill and zooplankton. All of them are right now on a boat, on a research vessel cruise going up and down the peninsula. They left from here 10 days ago and they don't get back until early February. I'm done.
Okay, Ms. Patnod's class, do you have I see you have someone ready with a question.
Go ahead, Natalie.
Is there phytoplankton in the snow?
Actually, there are, and they call them ice algae instead of phytoplankton. But they're very similar, and they're or snow algae, and we have them in Maine as well. Sometimes you'll be in the winter and springtime, and you'll see that the snow is a little bit pink, not where somebody has poured some juice or something like that, but naturally. And so those are snow algae, freshwater algae, instead of phytoplankton. Over here, if it's marine phytoplankton, there is also freshwater phytoplankton. Next question.
Okay, Ms. Wester's class, do you have a question?
Yes, I do.
Yes.
How do phytoplankton eat? How do phytoplankton?
The question is how phytoplankton eat?
Yeah.
Yeah.
Okay, so phytoplankton, I'm going to use the word quote, eat, just like plants on land eat. So they eat, or they take up sunlight in order to photosynthesize and produce organic matter. They also take CO2 from the air, and then they take, in the phytoplankton, the marine phytoplankton, takes CO2 from the water. And then terrestrial plants take nutrients from the soil or from the water, right? Many of you will add or have seen your parents add plant food to your plants at home. Here, the nutrients are in the water, especially in the ocean. And so they take those three, and with those three, CO2, sunlight, and nutrients, is that they grow. That's how they grow. It's called photosynthesis. Next question.
Okay. Ms. Train's class, do you have a question?
How fast do phytoplankton grow?
That's an excellent question. So some of them grow very fast in the sense that they can double, in other words, divide once per day. That's really fast. Think about that you would grow to twice your size or height in one day. Most of them grow less than that, maybe a fifth of that. And so they call that slope 0.2. And that's exactly what we're doing in our container, is we regulate the amount of food that we give them such that they grow at two different rates. And one group is growing slowly, and one group is growing fast. And we want to know, if the phytoplankton grow fast, do the bacteria respond fast? And do the genes get turned on fast? Next question.
Okay, we'll keep going until maybe for a few more minutes. So let's see, let's go back down. Okay, I see Dedham left us. So why don't we go back down to Ms. Venger's class? Do you have another classroom?
Yes.
Another question?
Nitrogen 7, what's your question?
What time zone are you using?
Lisa, can you repeat the question? What time?
What time zone are you in? What time zone?
Oh, I am in the same time zone as in Chile. So it is 2 hours later here than in Maine.
So that means it's about just after 10 o'clock here. So it's just after noon down where Patty is. All right, good question. Ms. Patnod's class, do you have another question?
We are good right now. Thank you.
Okie dokie. How about Ms. Westra's class? Do you have another question?
You can ask.
How long have you been researching phytoplankton?
I have been researching phytoplankton for over 30 years. And so after I finished high school, I went to college and I majored in marine biology. And already then I was looking at phytoplankton and chemistry, nutrients. How do they grow in upwelling regions? Then I went to get a master's and a PhD, and for there, first I looked at the diversity of phytoplankton, how many different species are in one part of the tropical ocean. And then I spent a lot of time in the microscope there, and I thought, it's beautiful, but I don't want to do this anymore. And so I returned to the interaction between phytoplankton and chemistry. Did a PhD at the University of California, San Diego, at Scripps Institution of Oceanography. And then in '89, I moved to the University of Miami in Miami, Florida. And I was there for about 9 years. And by now I was moving to look at phytoplankton, the production of gases and things that relate to climate and clouds in phytoplankton. And that's a topic for another phone call. And then in 1995, I moved to Maine, to Boothbay, to Bigelow. And I've been there for the past 20 plus years studying phytoplankton, but mostly in the Arctic, not in the Antarctic. Next question.
All right. Ms. Train's class, do you have another question?
What do you have another question? How long did it take you to get there?
From my house, the minute I left my house to when I got off the boat here at Palmer Station, it took a week, 7 days.
Excellent.
Next question.
All right, so let's see. Ms. Venger's class, do you have another question? We'll make this we're gonna make this our last round of questions because it's almost 10:15.
What types of areas do you catch your phytoplankton in?
Can you repeat that, Lisa, for me?
Can you repeat the question?
What types of areas do you catch your phytoplankton in?
Yeah, you said you pick them 5 meters deep, but where do you go out to go get the phytoplankton?
Oh, okay. I'm going to turn this thing so that you can see out, okay, out of the harbor here. So there is an area that within which we are allowed to sample. Remember, I said that Antarctica is a protected area? And so within this area, which is a little bit further, you see these islands and those icebergs in the distance. There are a couple stations that we visit all the time. One is Station B, which is sort of off that island black thing that you see in the distance. And another one is Station E, which you don't, which is about 2 nautical miles to my left here, which is very deep and does not have as much influence from the glacier melt. That's very important because when the glacier melts, you have fresh water that goes on top of the salt water. And not all phytoplankton like it, but mainly it's not representative of the open waters around Antarctica. And we want our results to be able to scale them up, generalize them to the open waters around Antarctica. So that's why we go out to Station E. It takes us about half an hour by one of these boats. Actually, we go with that. Oh, it's very dark for me here to see, but there is, let me see.
The lighting's pretty good for us, Patty.
Oh yeah, so you see there's a metal boat with 2 white bumpers, yeah?
On the
So
left
we
side
call
of the
that a
screen.
landing. Yeah, we call that a landing craft because it allows us to take an instrument called a rosette. It has 6 cylinders of 4 liters each, so we can take 24 liters of water at once instead of putting one down at a time. Having said that, is very flat, and so it just dances on the water, so it's not very stable, not as stable as the Zodiacs are. But we take that, it takes us about 20 minutes to get to Station E. We do our sampling, it takes us about 3 hours to collect the hundreds of liters that we need to bring back. And so we do this over 2 days. One day we go out, we bring water, we filter it, it's going to be our food. Then we go back the next day. Those are going to be our samples. And we use 5 meters because we need to match the light that we have inside the incubator. And we also, yeah, otherwise there's so much ultraviolet radiation, UV here, that if you expose them to too much, just like we get sunburned, right? If we go to the beach and you don't put sunblock on, well, the same thing happens to the phytoplankton. They get bleached. And so then that's not normal either. Next question.
Ms. Patnod's class, are you done with questions?
Yes, we're done. Thank you.
Okay, thanks. Ms. Westra's class?
We're
more
done. We're done. Thank you.
And Ms. Train's class, are you done with questions?
I think we're done, but we're
Okay. All right. Well, thank you everybody for joining us today. And thank you to Patty for speaking to us from Antarctica. It has been wonderful seeing you.
Oh, wonderful. It was my pleasure to talk with all of you. And you know that if you still have questions, because there were many more, for example, that came to me that I didn't have a chance, do put them into the WeatherBlur website. And I'll try to answer some of the ones on there, the ones that I didn't get to answer now. Okay?
Excellent. Thank you very much, everybody.
All right.
Have a great day.
Guys.
Wonderful.
Lisa, I have a quick question before you sign on.
Thank you.
Thank you.
Bye, everybody.
Bye.
Marcy, you had a question for me, you said?
Yeah, we're having trouble because we tried to put our observations of the plankton study on the WeatherBlur, and we had trouble with that. I don't know, is there something funky going on, or?
What were you trying to do, and what was the issue?
Well, every time I tried to post, or kid tried to post, it would say it's temporarily having difficulties or something.
So I've seen when was this?
I put some stuff up last night that the kids had written up.
And did you have trouble putting it up?
Yes.
Bye, Patty. Thanks.
Bye.
We did.
See you in Maine.
Yes. Have a safe trip back. So Marcy, I'm thinking they're doing some work on the WeatherBlur website during January and February. That's why we're not doing an implementation now. So that could be it, but I'm going to have to ask Gary. He might be able to. So this was last night you were putting stuff on?
Yeah, because the kids were fine doing their summary of the whole thing.
Okay. And you had trouble, and you got that message saying it was temporarily unavailable?
Yeah, or
Okay.
something it wasn't able to post.
So another thing I always warn people, because I've done this, make sure you're logged in, because you'll sometimes get a message like that. If you go to the site and you click to download data and you're like, oh wait, I didn't, it'll actually act like you're uploading the data, but then when you click submit, it gets you a message, and I've done that before. I went, oh, now I have to
I
log
forgot
in.
you said that, and I didn't check that, so I will do that and see if
Yeah.
I can get all of the stuff with handwritten in there.
Yeah, all right. If you are logged in and you have trouble, let us know, because it might be that they're working on the website. We've given them a big list of stuff we want them to take care of by the end of the month. So that's also another possibility.
Okay. Also, did you record this?
I recorded half of it. I forgot the first half. I was so bummed. I was looking and going, I'm like, oh no, I forgot to record. So I caught the second half when she was mostly in the lab. I caught her just before she went into the lab.
Because that might be cool to look back at, or to let parents look at.
Yes, it will be.
All right. Well, thank you.